So now I'd like to introduce Ian Mellis. Ian comes to us from Columbia University, and today he'll be telling us about transcriptional adaptation.
Thanks, everyone. Thanks to the organizers for the opportunity to present. So I am going to tell you all about the underlying substrate now of perturbations and a mechanism that may complicate the way that we interpret different types of genetic perturbations, following up on some of the excellent talks we just heard. So transcriptional adaptation is something that will affect the way that we interpret genetic perturbations and genetic perturbation screens that are done for a variety of purposes. This is just a review of the field. It's intentionally a dense slide. Perhaps of most interest to this audience is using genetic perturbations to understand fundamental aspects of molecular and cellular biology, including inferring gene regulatory network structures. But it's also used throughout biomedical research to study drivers of phenotypes of interest across cancer contexts, infections, medical genetic disorders, immunology, and many, many more fields. And there are a variety of underlying mechanisms and methods that are used to conduct genetic perturbations. As Tom described earlier today, across screening methods, there are some really wonderful CRISPR-based methods that I have here from a review on the screen. We won't discuss all of them in detail, but to say that on the top left, what you see is the classic CRISPR-Cas9-based knockout method, causing indel mutations in a gene of interest, theoretically knocking out most of the alleles that are affected. On the lower row, what you see are a variety of CRISPR nuclease dead conjugated protein constructs that change the way that genes are expressed. They either interfere with gene expression through CRISPR-I or related methods, or they activate gene expression through a variety of methods. The classic methods for genetic perturbations actually, in addition to genetic knockouts at the DNA level, for screening purposes, often relied on RNA targeting. And so in the literature, for the most part, it's prior to the advent of CRISPR or the discovery of it, there were shRNA screens, siRNA arrayed screens. Morpholinos were used as an RNA targeting method. At the protein level, Degron tags were used to modulate protein expression on demand. And there are many, many more methods. All of these are genetic perturbation methods that unfortunately can casually be conflated with each other, because the thought is, I just want to knock out or knock down my target of interest and see what happens. That's the perturbation. What's the output? But they're not the same. And I'm going to tell you why specifically genetic knockouts that cause nonsense mutations are definitely not the same in a lot of situations compared to knockdowns, even super high efficiency knockdowns. And the implications for that are, I think, pretty widespread in our interpretation of our experiments. So we've known for decades that there are often discrepancies between knockdowns and knockouts of the same gene. And this is just one example that really led the authors of one of these papers in 2019 to discover transcriptional adaptation. And this is zebrafish development. This is 3.5 days post-fertilization of zebrafish embryos. The brown blob in the middle of each panel is the developing zebrafish liver. And in the wild type, in the left panel, you see a nice, robust liver. When you knock down CAPN3A, which is a critical regulator of liver development in zebrafish, using a morpholino targeting the RNA of CAPN3A, you get a lethal phenotype with very little liver development. The authors also came across an allele that was a frame-shifted mutant of CAPN3A. It caused a frame shift very early in the gene, which led to a nonsense mutation in the first few dozen amino acids. And instead of seeing a dramatic loss of liver development like they saw at the morpholino, they saw perfect robustness. And these zebrafish developed normally. Now, normally in the literature, if you remember from microRNA, the discovery and characterization of all the targeting rules of microRNAs, everyone was talking about off-target effects of RNA targeting genetic perturbations. The authors did extensive experiments to show that this was not an off-target effect of the morpholino that led to loss of liver development. It was in fact the case that there was a difference between knocking down the RNA and getting a very good knockout of the gene, leading to differences in the phenotype. What they and another group simultaneously published in the same year was the discovery of a phenomenon, not necessarily a mechanism, called transcriptional adaptation, which acts at the level of mutant transcribed RNA that is degraded, somehow shuttled back into the nucleus, and leads to semi-targeted upregulation of partially homologous genes, which sometimes compensate functionally. And this is critical to further characterize if it's true in the context that we are conducting genetic perturbations, because this potentially explains many differences between knockouts and knockdowns. If the knockdown method does not act through the same mutant RNA decay pathways that affect, say, nonsense transcripts. So Yogesh and I read these papers, and we were really interested in understanding this phenomenon more. And so we asked the question, does transcriptional adaptation occur more broadly than in just zebrafish embryo development? In one of the papers, there was some suggestion that it may occur in one context in mouse embryonic stem cells, but really it had not been characterized beyond a handful of carefully curated genes in zebrafish development beyond that. And so we wanted to characterize it more broadly in mammalian systems in particular, so mouse and human systems. And we wanted to ask, does it matter in those systems as well, phenotypically, potentially? And this work is really a close collaboration between Yogesh's group and two wonderful grad students who worked with him, Madeline Meltzer and Nico Bodkin. So our contribution to this effort was having the unfortunate insight that a lot of our methods may be causing something that we didn't know existed, which is that our insight was that CRISPR-Cas9 may lead to transcriptional adaptation, we think, because it causes nonsense mutations inherently. It's causing Cas9 nuclease activity leads to double-stranded DNA breaks, which lead to indels, which 2/3 of the time at least lead to frame shift and nonsense. Therefore, we combed through publicly available data for all paired CRISPR-Cas9 mutagenesis RNA-seq data sets that we could find in bulk RNA sequencing. And we looked for evidence of transcriptional adaptation, not definitive conclusion of transcriptional adaptation, but signs of it. So for a given reference gene that's targeted by Cas9, we looked at its annotated paralogs. And we asked, after mutation compared to the control, is there upregulation of the paralog? That's it. We didn't care about anything else in these RNA-seq data sets. We were just repurposing them outside of the underlying biological question that they were originally published for, to combine these data sets together and answer the question, are we seeing transcriptional adaptation broadly across the genome and across many biological contexts? So GRINE is a tool that is sort of a subset of the gene expression omnibus, where they consistently quality control and map and count RNA-seq data for human, mouse, and rat data sets pulled from GEO. They have thousands of data sets. We quality controlled experimental designs that used CRISPR-Cas9 in particular, including with bulk RNA sequencing data. We found over 200 experiments that we personally reviewed all of the experimental designs and quality control to make sure there were adequate controls, adequately homogeneous cell samples, that we weren't getting really differences in cell type compositions confounding our interpretations here. We annotated all the paralogs just by lookup through Ensemble. We quality controlled and processed the data sets consistently with each other, hand annotated all of the control samples and compared those against the published metadata. We conducted differential expression analysis, and we developed a bootstrapping pipeline for estimating the significance of paralog upregulation above random expectation. Because as the discussion earlier alluded to, you get a lot of dysregulated genes in some cases, especially in bulk RNA sequencing data sets. And so why wouldn't you sometimes have paralogs that are upregulated among the hundreds or thousands of genes that are upregulated after any random genetic perturbation? And so we developed a statistical pipeline based on underlying baseline gene expression for answering the question of how significant this is. Yeah.
But don't you want to do it with CRISPR-I at the same time?
Ideally. So that's exactly right.
Even if they're not matched in the exact same study, you could at least run the analysis, but on CRISPR-I experiments instead of CRISPR knockout.
Yeah, so one thing I'm not saying is that the effect sizes are very small in genetic perturbation, in transcriptional adaptation. And there's going to be a lot of batch-to-batch variability. And additionally, we think that the underlying biological context will have a lot of effect on the magnitudes of the changes. So across laboratory, across user, across cell system, differences would probably confound substantially any matching of data sets that aren't conducted consistently. There's one unpublished data set that we have not generated that's coming out soon that will address this question. But we agree with you. This is, as a junior, PIO is not going to be generating these massive data sets specifically to answer this question on spec. I wanted to.
There is a paper out from Jonathan Weisman's group on exactly the same experiment design just 2 months ago.
Yeah. And
the same screen, CRISPR-I, CRISPR-N, and found some differences.
I mean, we have it. We've published the exact same
thing.
Panel of genes, CRISPR-I, CRISPR knockout in the same cells.
Oh, you have?
Yeah.
Okay. I'll definitely read into that. Sorry, I didn't see that before.
Don't let it happen again.
I won't. You can count on it.
Missing something.
Yeah.
Really transcriptional reasons why you get upregulation of paralog genes.
There are many other reasons that could explain.
How do you distinguish that given this?
We're not. That's why I'm saying this is evidence suggestive of transcriptional adaptation that needs to be confirmed by comparison with other knockout methods, knockdown methods, protein targeting methods, all simultaneously showing that it's only mutant RNA causing upregulation. But yes, it's more evidence of genetic compensation generally rather than, say, transcriptional adaptation. Long story short, we found that more than a fifth of the analyzed CRISPR-Cas9 targets had more paralogs upregulated than would be expected by random chance. On the x-axis, what you're seeing is the fraction for each dot. Each dot is a CRISPR-Cas9 target. And on the x-axis is the fraction of its annotated paralogs that get upregulated after Cas9 targeting. On the y-axis is the p-value, negative log 10 p-value transformed, showing that we see a lot. We then asked, might this be important? And particularly, might this be important in the context of gene regulatory network inference? So we asked for the targeted genes that were transcription factors, the downstream genes that they regulate, and that their paralogs are known to regulate as well based on annotated regulons. How robust are those downstream gene expression levels to mutation of their upstream regulator? And we would expect that if there is robustness, there would be a very minimal change in gene expression up or down of the downstream gene after the upstream regulator is mutated. And if there is no adaptation and there is no robustness, then the downstream genes would go wildly all over the place. And sure enough, what we see on the right panel on the x-axis, there are two groups for downstream targets of transcription factors that we saw no paralog upregulation for on the left. On the y-axis, you see the downstream gene expression changes, and you see that they go all over the place. But for transcription factors that do demonstrate adaptation or paralog upregulation, their downstream genes are very tightly controlled after knockout, suggesting that there is some sort of stabilization of their downstream gene regulatory network output. So with that, we wondered what regulatory relationships may constrain transcriptional adaptation. When does transcriptional adaptation work? And so we turned to stochastic simulations to simulate toy gene regulatory networks that may demonstrate transcriptional adaptation. And so in this setup, we have a transcription factor in dark gray that eventually will get mutated. Its paralog transcription factor in light gray, both of which regulate the expression of a downstream effector or downstream target gene that may be important for development, whatever, in orange. And this is a bursty transcription model with two independently bursting alleles. And I'd be happy to discuss the details of the setup here. There are 8 free parameters, all scaled to the degradation rate. And we can discuss the details separately. We scanned over tens of thousands of different parameter sets. And this is just a representative example of some of the output. And on the top panels, what you're seeing are traces of gene expression under a single, each panel is a single parameter set. And in gray are the gene expression levels over time for the transcription factors, in orange for the downstream target gene. And as has been shown before in the literature, over time you can over sufficiently long periods of time, you can condense these to independent pseudo-single cell samples at single snapshots in time, very similar to what you might get in a single cell RNA sequencing data set and gene expression levels. And you can effectively summarize them as distributions of single cells under the same gene regulatory network control. And we can recapitulate differences that are seen in real data between gene expression distribution shapes in single cell data sets, where you have a vaguely symmetric unimodal distribution of downstream gene expression under one parameter regime and a bimodal gene expression distribution under another parameter set. We classify downstream network output as robust or not robust to upstream transcription factor mutations based on their single cell gene expression distributions. And I just colored these gray for visibility because it doesn't look as good with orange. We looked at robustness both at the level of average expression level and in distribution shape, and in both distribution shape and mean. Using decision trees across these many parameters sets that we scanned over, we found that there were only a subset of parameters which were highly related to transcriptional adaptation like relationships in our network setup that were important for downstream effector gene robustness to transcription factor mutation. And so what you're seeing on the left panel is this subset, this subsampled space that we identified via decision trees. If you resample only within that subspace of the parameter sets, you get on the right panel way more robust downstream gene expression level distributions than you get if you just sample across the entire set of the entire parameter space. So in conclusion, we found that transcriptional adaptation, or at least genetic compensation of some sort via any mechanism, may be widespread in mammals, and it's probably present across many biological contexts. Because we harvested RNA-seq data sets, and I didn't discuss, we did a single cell RNA-seq data set analysis as well to show that we could get transcriptional adaptation observations at the level of single cell RNA-seq data across many biological contexts: cancer in vitro models, models of development, physiology of many different of many different tissues and disease states. And in many different cases, we observed some evidence of genetic compensation. However, this is likely not ubiquitous. Why is it not ubiquitous? Why does it occur? Are many outstanding questions, which we'll get to in a second. Gene regulatory network stability, at least at the level of annotated regulons, is associated with transcriptional adaptation or genetic compensation, at least in our analysis. And only a subset of the modeled regulatory relationships appeared to confer robustness on gene regulatory network outputs in stochastic simulations. So there are a lot of implications for future work. The top level point I want you all to take home is that transcriptional adaptation inducing mutations can affect how you interpret Cas9 or other mutation-based knockouts compared to CRISPR-I or other knockdown-based genetic perturbations. We think that the importance of transcriptional adaptation is going to depend on context, and that context can be of many types of contexts. It's going to be in different biological contexts, in different gene regulatory network structures, and there's going to be a lot of work to do on understanding when it matters. We're going to need to do these systematic comparisons of mutation-based genetic perturbations, chromatin targeting, transcript targeting, protein targeting perturbations to really narrow down where transcriptional adaptation specifically is occurring, as opposed to feedback loops, as opposed to other mechanisms of compensation, in order to understand what we're dealing with as the biological substrate of our perturbations. And the rules themselves for when transcriptional adaptation is working need to be clarified. When is a mutation going to trigger upregulation of some partially homologous genes? Why are some paralogs upregulated and not others? Which mutations are most likely to cause upregulation of paralogous genes? And many related questions. So there are a lot of outstanding questions that I think would be great to discuss over coffee. With that, I just want to make sure that I acknowledge everyone and all the funders, and just let you know that I'm recruiting postdocs. So if anyone's interested in this or virology or antiviral immunity, please let me know. Leo?
Well, yeah, I'll ask my quick question and then clap. I will clap because this is very nice. But I'm almost convinced, but I have a question about the bioinformatics of quantifying the pseudogenes, because they're notoriously really hard to get right. And you might imagine that, you know, the nonsense may have sent reads like to a pseudogene that maybe were there to begin with, but then incorrectly mapped previously. So how are you confident that you're getting the quantification correctly?
That's absolutely a great point. We are relying on Ensemble's annotation of paralogs, not pseudogenes.
I'm sorry, I said pseudogenes, but I meant paralogs.
Yeah, no, they're equally problematic. There's a variety, there's a whole distribution of levels of homology among different families of paralogous genes just based on the gene tree itself and how it relates to other organisms, of course. And so each gene family will have different levels of homology among the paralogs within it. We don't have a great sense. We've done the analysis led by Madeline about levels of homology versus the frequency of upregulation of these paralogs. And we actually don't see a great quantitative trend at the level of average homology across the transcript. Sub-gene level homology to be determined.
I just wondered about the sort of downstream implications, not just in terms of interpreting, but in terms of sort of engineering, modifications or interventions that might take advantage of TA. Like, I don't know that much about this. This is my introduction. So are people trying to actually use that sort of?
Yes,
already? Is that?
Absolutely. So what Yogesh referred to is a paper where they showed that you could use this for engineering. You can use transcriptional adaptation for engineering. You take a highly transcriptional adaptation prone transcript and you prepend a homologous partial subsequence of genes you're interested in upregulating, and you can trigger upregulation of some of those genes. And so for gene therapy, a lot of what I do is engineering gene therapies. We're really interested in using this system because it's entirely human protein components. There's no bacterial Cas protein that you're throwing into human cells in an already diseased state. It would be great if we could make this target, and it would improve on, as Tom was alluding to, CRISPR-A is really hard to get right. If this could be a better version, that would be
That's
great.
also part of Madeline's thesis.
Yes.
Oh, okay. I'll
All right. Thank you all. Oh, sorry.
Well, I was just going to ask, you can assess this for the things that are direct paralogs, because then you sort of know where to look. But what about off-target TA? How much circumstantial homology is there out there? And do you see? Yes.
We are very interested in that question. What are the targeting rules? It's not just paralogs. In the original papers, they actually used any blasted subsequence as a search function for genes that might be affected by transcriptional adaptation. They got a lot more results. It requires a lot of ad hoc tuning to do that. And fundamentally, we need to understand the targeting rules. And then we can get a sense of what off-target effects, what maladaptive transcriptional adaptation might be in that case, because there are a lot of contexts where there are nonsense mutations, there are bad effects. Are those a result of the underlying mutant gene, or are those a result of some maladaptive compensation? Sure.
So have you seen any effect on the cofactors? Are they showing any evidence of adaptations as well for the transcription factor cofactors?
We haven't looked at that, but that's a great thing that we should look at.